Welcome to my poster. My name is Carlus Deneke. I'm from the German Federal Institute for Risk Assessment, based in Berlin and abbreviated BFR. And I worked as a bioinformatician supporting the national reference laboratories for different food pathogens. And what I want to present here is how we interpret or provide bioinformatic pipelines for the easy interpretation of genomic data. So we receive at our institute a large number, or a fairly large number of isolates, so several thousand isolates per year, which are being sequenced at our institute. And my and the job of my colleagues is to provide efficient and fast analysis to the users from the individual laboratories. So we do this by a set of pipelines that I want to briefly present here, so which are called AQUAMIS for quality control, Bacharach for characterization, and SNAKE for CG-MST. I want to begin with the first pipeline called AQUAMIS for assembly and quality control, which is a snake-make pipeline, so it connects different tools that are known in the community, and they're connected, and the ultimate goal is to connect a number of tools and present the users in the different laboratories with an easy to interpret interactive report, so that they shortly after sequencing can make an informed decision about the quality of each of their samples. So one of the key features of this pipeline is that we perform something that we call auto-QC. So each of the different tools performs different analysis and outputs. And in the end, we have certain outputs, so-called QC metrics, combined with a number of thresholds, which can be species-specific or also species-agnostic, so that in the end, we have for different QC metrics, for example, the Q30, the coverage depth, or the assembly length, a flagging system in green for pass, orange for warning, and red for fail. And this is displayed here in this chart, where we have for each sample a row and the different metrics, and they are colored. And in the end, when there's one QC metric that fails, that is red, the entire sample is failed. On the other hand, if there's no failing flag, the entire sample is estimated as pass. And this has now the advantage or the application that we have a lot of samples. Say you sequence 100 samples at a time, and typically 98 samples, for instance, do not fail, so they have a pass, so that the users can dedicate more time on the corner cases, the failed cases, and possibly overturn the decision or restart the sequencing of those samples. So this is the first pipeline, the so-called AQUAMIS pipeline, which is automated in application, and users usually, after a few hours after sequencing, or up to 1 day, receive the quality results. But since it's automated and the preliminary QC decision, the auto-QC is also automated, it also means that the assemblies generated with the pipeline can be used directly in other pipelines, namely in the characterization pipeline, Bacharach, and the SNAKE pipeline, CG-MST. So also these pipelines are so-called snake-make pipelines. So we take advantage of snake-make here, that we, on the one hand, can combine a large number of tools, but that also snake-make recognizes automatically for which samples result exists already and for which no results are present. So that means when I have a collection of samples, say 1,000, and I add 20 newly sequenced samples, which come from the AQUAMIS workflow, snake-make recognizes that I only need to compute 20 new results, and I can combine this with the results from the 980 remaining already existing results. And so the Bacharach pipeline, as I said, is meant for characterization, so it does fairly normal things like looking at the rhizostome by searching for AMR genes, serotyping for a number of species, reference genome finding. It tries to estimate which contexts belong to plasmids. It also looks at virulence genes and so on. So it has a number of modules. Some of them are species specific, some of them again agnostic and work for all kind of bacterial species. In the end, we have the results, a large number of results of different tools which are combined and reported, and sometimes just the combination is of interest. For example, whether a particular AMR gene is predicted to be on a plasmid or not, because it has different implications for risk assessment. And again, the output of this pipeline is a report, so the users can actually look at the reports. And at the same time, for all pipelines, we try to design so-called JSON documents, so that we have also the results stored in a computer-readable format. Yes, so much about the Bacharach pipeline. Now I want to finally talk about the Chewy snake pipeline. So Chewy snake uses as the core Chewbacca, hence the name, as the core module for allele calling. So it's a allele calling pipeline which is mainly built on Chewbacca. And at the beginning, again, for each sample that is part of this workflow, that was added to the workflow, it performs allele calling. So we receive the allele profile. And then, in turn, we do a large or a number of steps to go from these allele profiles to informed information about the clustering and the relationship between different samples. So I want to briefly go through the steps of this pipeline, and then later also explain how it is applied. So after the allele profiling using Chewbacca, we convert the resulting allele numbers in so-called allele hashes. I will talk about this in a second. Then we have options for allele filtering, so to exclude certain alleles that, for example, contain frame shifts. Then from the set of all allele profiles, we want to infer the distance between all samples, the pairwise distance, and for this we use the tool GrapeTree. GrapeTree at the same time constructs a minimum spanning tree, so that we have a form to visualize the entire connectivity of all samples, and this can be actually loaded into the GrapeTree web interface. So after that, it is also convenient to place each sample into a cluster, and we do this by using single linkage clustering and dividing at different thresholds of different allele thresholds. For example, large ones of 1,000 to loosely group the samples, but also small ones like 5 allele differences or 10 allele differences to group them in putative outbreak clusters. So this is done by the clustering, and as before, all this information is collected and presented in an HTML report, which also informs about allele quality control, so that end users have at a single document in an interactive way all information necessary for making a decision about outbreak relatedness or not. I just want to briefly sketch two things that are relevant here. So I mentioned that we hash the alleles, so what is the idea behind this, and why do we want to do that? So when you do allele calling, you have the situation that you find new alleles that have not been discovered before, and any allele caller will assign a new allele number to your allele. And if you use a system like a platform which does the allele calling for you, for example Enterobase, of course all users of the Enterobase platform will use the same scheme with the same allele numbers. But if you use an offline command line tool, the allele number that is assigned for your sample is not necessarily the same as the allele number that is discovered at a different place. So this brings upon the problem of that you're not synchronized with others. So of course one could synchronize and think of nomenclature server systems, but what we designed here is a way that circumvents this problem in the sense that each of the alleles that is discovered is hashed using a hash function. And so we have a recipe of naming alleles in a standardized way, and that means whoever uses this pipeline locally will, whenever they discover an allele sequence, will get the same allele number. So that means independent discoveries of novel alleles automatically receive the same allele hash. And that means that the allele profiles that contain allele hashes can be directly compared. And this opens the way to exchange allele profiles between different partners, from partner to partner, in a peer-to-peer fashion, for example. And you don't need a big system where everyone exchanges the data. It is also possible that, for example, it reduces the amount of data that needs to be exchanged, because if different laboratories agree on the same procedure to name the alleles, namely the hashing, it suffices to just exchange allele profiles, and thus there's no need to reproduce the entire analysis again, for example, by exchanging the raw FASTQ data and redoing an analysis. So that means once I do locally, like right after sequencing my analysis, I can send out the analysis results, and they're comparable with others. So I send them, and they can make a decision on relatedness. And this is also illustrated here in this lower part, which describes the so-called Chewy join workflow, where data from different laboratories is joined. Also here, a distance matrix is created, and clustering and cluster naming is performed, and again, an overview is given. So this summarizes the Chewy snake pipeline and all the pipelines I wanted to present. What I wanted to say at the end, the pipelines are all openly available on GitLab. You can find the address here, and they are also available in Bioconda and as Docker containers. And yeah, I would be pleased if you're interested in the pipelines, trying them out, and contact me if you have questions. Thank you.